Bioorthogonal Chemistry, thank Carolyn Bertozzi. Also congratulate her on that Nobel Prize in Chemistry while you were at it.
Bioorthogonal chemistry is kind of like chemistry and biology that play nice with one another. So it takes the versatility of chemistry, mixes it with the specificity of biology, and allows you to add custom modifications to biological molecules like proteins and nucleic acids, so like DNA and RNA and glycans, so sugars, and lipids and all sorts of different biological molecules by taking advantage of some of the best of both worlds. So when I say chemistry is versatile, there are all sorts of different reactions that you can do. You can synthesize all sorts of different molecules in the lab, things like dyes, fluorophores, the things that will shine light, various like chemical handles that will allow you to purify things out of cells, all sorts of different things that you could do with chemical reactions. But the problem is they're not very specific typically. When these reactions occur in the lab, we have to go through all sorts of different processes in order to prevent them from interacting at places that we don't want. This typically involves the addition of things called protecting groups, which basically hide the parts that you don't want to react yet, so that you're only reacting at specific positions. So for example, when we want to make nucleic acids, so like chains of DNA or RNA in a lab, you have to go through all of these different cycles where you're protecting different groups so that the reactions don't occur. In our body, however, this is carried out by specific enzymes. So these reaction helpers that actually hold the specific molecules in specific positions and add these modifications or add the partners directly in the positions. But this is at the expense of versatility. So this is super duper specific, but it's not very versatile. So these enzymes, these reaction helpers, speeder-uppers, they basically are very specialized for adding specific things in specific locations on specific molecules. And so this is really great for making sure that you don't have unwanted reactions going off in your cells. But if you want to add something that your cells normally wouldn't have, if you wanted to add, say, a fluorescent molecule so that you could track where various molecules are, where modifications are being made, well, then this isn't going to be possible just using the cell's own machinery, especially if those groups that you want to add are maybe they're toxic to the cells or they're really, really big and bulky. And so adding them is either impractical or it would interfere with the cell's normal processes, and therefore it wouldn't be a very good like readout of what you want when you're trying to see what's actually going on. And so bioorthogonal chemistry is the idea that you can introduce kind of precursors to that final modified product that you want, get the cell's own machinery to incorporate those precursors. So these precursors are like modified versions of the building blocks, not of like the final product that you want. So it's not like you're adding a full modified protein into cells. Instead, maybe you're adding a modified nucleic acid, or a modified amino acid, so one of the protein letters. There are various tricks that you could use to introduce these at specific locations, such as the incorporation of non-canonical amino acids, which we'll talk about more in another post, but basically it's this idea that you can kind of engineer the genetic instructions for a specific protein so that you can have it incorporate a protein letter that isn't normally there. There are also other techniques that you could use to introduce such amino acids. I'm not going to go into them here. You can introduce these modifications to other molecules as well. I'll get into some of the work that Carolyn Bertozzi did with glycans, so with sugars, where she introduced precursors to these sugars. And then these precursors got incorporated into these complex carbohydrates. So basically these chains of sugars that cells decorate their surfaces with. And she wanted to track where these various sugars were. And so she was able to feed these cells this precursor that then later she could react with these chemicals. And in the chemical reaction, it would introduce a fluorescent molecule, and then she could track the cells. She also had versions where she could then like target the cells for for destruction. And this was helpful because these cells, sometimes in like various cancers, the surface chemistries of like the glycan chemistries, so the various sugars they display, can actually change during cancer and sometimes be overexpressed, so more than usual and that sort of thing. And she's actually working to develop therapeutics that can target the sugars. And this was helped out by being able to track these sugars using this sort of bioorthogonal chemistry. So the key concepts are that the biology is going to be dictating where that reactive group is placed, and so where you want the modification to occur. And then the chemistry is going to dictate what modification you're going to add. Often what's happening is there's some sort of click reaction. So click reaction takes us to the work of two of the Nobel laureates for this year, Morton Nobel and Barry Sharpless. And so basically the idea with a click reaction is that you have this simple chemical reaction with two reactive groups. That's really specific. So it takes advantage of specificity by incorporating a reactive groups exactly where you want them. And this reaction is quick, it's versatile. You can basically stick these things onto various molecules, kind of like if you were to stick belt buckles onto various things. And so this is from the Nobel Prize announcement. I'll show you later. They have some really nice educational content that you can read. They have like a popular science version and like a more detailed technical version. But you have this basic reaction. This is kind of like the canonical click reaction where you have this azide, so these like three nitrogens, this alkyne, this like triple bond to a carbon. And then they discovered that you can actually catalyze this reaction, so speed it up and get it to be really, really fast and efficient by adding copper. And this promotes the cyclization, which then actually forms a bond between these two molecules. And this reaction has subsequently been modified in various ways, incorporated into all sorts of molecules, and it's super duper versatile and it's used in a ton of different industries. And this is why these two were awarded the Nobel Prize. But Carolyn Bertozzi wanted to take things a step further because she wanted to actually use these inside of cells and inside of organisms. And the problem was that the biology or the chemistry of these things wasn't very compatible with, well, like life. So one thing is that there's this copper molecule, and copper is toxic to our cells in our bodies. We use a tiny bit of copper, and there are some very important enzymes in our bodies that rely on copper. But what happens is that within our bodies, this copper is actually chaperoned wherever it goes. So there's proteins that hold tight around this copper and take it from place to place. And there's actually like copper storage diseases where you have excess or too little of copper or improper transport or storage of copper, and these can cause really big problems. So in order to actually get this reaction to occur efficiently, because you need the copper for it to be really efficient and fast and all this stuff, Bertozzi had to figure out a way to make it happen without copper. But I'm getting a little ahead of myself. And so let's step back a second and talk about just what the idea of bioorthogonality is. So this is a really great review article. It's fairly technical, but it also has just a basic background. And then it gets into technical details as well as a lot of great examples and citations and links. And so I'm going to provide a lot of links to various resources. I don't have time and I'm not the best person to write out a detailed blog post on this, but I will point you to these resources. So this review article is really, really great, and I highly recommend it. According to them, so a bioorthogonal, there's various things that have to be met in order for a reaction to be considered bioorthogonal, a term that is attributed to Bertozzi in her lab. So the reaction has to occur at temperatures and pH of physiological environments. And so this is a really, really important one because those chemical reactions, the ones that actually happen in the lab, when these are being done in the lab, they're typically being done under very harsh conditions. And conditions that would kill cells, and that wouldn't be good. So even if you don't need protecting groups in order to hide the parts you don't want to react, you still have the problem that in order to get these reactions to occur, you're often using conditions that are very far from physiological, so very far from what's like actually natural, and therefore you would be messing up all sorts of things, if not outright killing the cell or organism. It also has to be selective, and in high yield. So you want to make sure that the modifications are only happening where you want them. And remember that we're not adding protective groups and stuff. And so we have to make sure that the modifications are going to be super duper specific, which is why when you add that original reactive group, this reactive group isn't one of the typical reactive groups that we see in biological molecules. So our biological molecules like proteins, nucleic acids, lipids, glycans, all these various things, they have functional groups that can serve as reactive molecules. So um, they have things like these amine groups, like these bio groups, hydroxyl groups, various groups that can interact. And in fact, they do interact with the help of enzymes. But remember, those enzymes are providing the specificity by binding onto the specific molecules, kind of like orienting them so that the reactive groups of various molecules are coming close together. When we're talking about these reactions occurring in chemistry in the absence of enzymes to actually dictate where the reaction occurs, well, now we need to make sure that those reactive groups are super duper specific for one another, and they react really fast, and irreversibly, and that they, to a really high degree of specificity and efficiency. And that's a lot to ask for. And so they have to have these reactive groups that aren't things that the bodies naturally use. And therefore, you're not going to have these reactions occurring at positions where they wouldn't, where there aren't the modifications already, where there isn't that incorporated precursor. Okay, and so as we'll talk about, there's going to be various strategies in order to kind of like do these things, starting with some of the classic ones of that Bertozzi developed, and then some further advances. So initially she started with like a Stoutinger ligation. Actually, before that she had like ketones and then Stoutinger, and then the copper click chemistry came into play, and then she developed a version that didn't need the copper, this like strain promoted click chemistry. And there's been all sorts of different advances and different uses, and I'll get more into this in a minute. But these groups that are being added are going to be things that aren't naturally found in our body. So you don't naturally find these sorts of molecules in biological molecules, but you can get the precursors incorporated. And because you can have the precursors incorporated using those enzymes, so using the biology's specific machinery to introduce these unnatural versions where they would normally put the natural versions. These reactive groups, these precursors that you're adding, the modifications, although they're not natural, they're typically really small. And so it's not like you're adding that whole extra molecule that you want to add on there. Instead, you're just adding a little linker that is going to allow it to latch on to react with whatever modified group that you want that has the matching part on the other end. And so these enzymes can incorporate this, and they can incorporate where they would normally add it. And this is going to allow you to see where various things would naturally occur. There are also tricks that you could do in various processes, some of them more like synthetic, some of them like more outside of the actual cellular context, but then you can like add it back in order to introduce mutations at specific locations. And you can also use like modifying enzymes to add these precursors molecules like after the fact. It sounds kind of weird, but because there are various enzymes that modify existing molecules, you can get those enzymes to potentially modify those existing molecules to add incorporating a modified version, and then a modified version of the modification. But then you still are doing that second step to introduce whatever sort of conjugate, so the thing that you want bound to it to be. So whether this is a dye, whether this is some sort of drug, whether this is, I don't know, all sorts of different things. that you could do in order to attach this. So you might think like, okay, well, if proteins, I've talked a lot in the past about how we can genetically encode like tags and stuff to put on the protein so that the proteins get made with like an extra tag to help us purify it. You can't do that with, can't add like a peptide tag onto nucleic acids based on just modifying genetics. And you can't add it onto sugars and you can't add it onto lipids just by telling the cells to do it. And so instead, we have to add it after the fact. So you can add these sort of like purification handles or these sort of labels to these molecules that you wouldn't normally be able to. And so that's one of the cool applications of bioorthogonal technology. And even for proteins, you can add those fluorescent tags and things, but those might be bulky. They might mess up with what the protein's naturally doing. And so by adding these bioorthogonal precursors, you can then get the cells to do whatever the heck they were going to do originally. And then you add this chemical after the fact. And because you're adding it after the fact, you can actually do harsher reactions as well, and things that wouldn't normally be compatible with life, but you can do it after you actually like cells are fixed or whatever. So there are various different things you can do in order to introduce more extreme things, as well as the kind of milder things that you can do while the cells are still alive. But anyway, I've gotten a little off track. So the reaction must not be affected by water or endogenous nucleophiles, electrophiles, reductants, or oxidants found in complex biological environments. So more on these various terms in other posts, but the basic idea is that your cells, because reactions have to occur in your cells, they often occur with these various nucleophiles, electrophiles. These are kind of like reactive groups. So as we talked about, our molecules naturally have these reactive groups, but our enzymes take advantage of them and really get them to interact in specific ways. Whereas we don't want them to be interacting with the molecules that we add. So by bioorthogonal, we have to have something that is actually going to not use the cell's natural ways of making reactions happen in terms of when we want the modification to occur. We want the cell's natural machinery to incorporate the precursors, but not to incorporate like the actual modification into the precursor or onto the precursor. So we put that precursor that's kind of like already made into the cells, and then get them to incorporate that and then modify that. But the only part that's actually using the biological machinery is to incorporate the precursor, not to incorporate like an actual the modification that you're going to add afterwards. And so we don't want that modification that we add afterwards to rely on a reactive group from a natural molecule, because natural molecules, the reactive groups that they have, are going to be found on a lot of other molecules. Another thing is that like water. And so water is a huge thing that is often avoided in organic chemistry because water is really reactive with various processes and it can interfere with various chemical processes. And therefore, in these reactions have to be kind of like unlike reactions that you would often see in an organic chemistry lab because they have to be taking place in the presence of water. So the reaction must be fast, even at low concentration. So you can't be adding a ton of these things. They're just going to kill the cells. And the reaction products must be stable. So a lot of the reactions that occur in our bodies are easily reversible. So they can go in both directions. And this is really important because it allows us to use like biosynthetic pathways to make and break molecules using the same enzymes, the same reaction helpers. And this is going to be really important in metabolism, so the making and breaking of molecules, because these pathways, if you need sugar, you can break down glycogen, so these big sugar chains. If you don't need sugar right now, you want to store that sugar, well, now you can take that glucose, so the individual sugar pieces, and put them into glycogen. And you can do so using a lot of the same enzymes. And so this reversibility is really important. But if you're wanting to add a tag or something, you don't want that to be easily reversible, because then your tag's going to fall off, your label's going to fall off. Say you have a fluorescent label and you want to add it to molecules, you're going to want it to be added to the molecules and stay on the molecules. You're not going to want this label just like coming off and floating around. And then you say, oh, look, my thing's right there. And your thing, like your thing isn't right there. Your thing is right there, and the label's just like floating around. So you don't want that. And yeah, as we mentioned, these functional groups are not naturally present in biological systems. So this functional group, that's just basically this idea where most of organic molecules, or I guess all of organic molecules, the kind of definition is that they're built on this carbon hydrogen skeleton, so this carbohydrate backbone. These basically, you could think of these hydrogens as kind of blah. And when you add something more reactive, you call this like a functional group. And various functional groups have different reactivities. They can react in different ways that can be taken advantage of by enzymes to make specific reactions occur. But when we're talking about bioorthogonal chemistry, because we want this chemistry to be happening kind of like in parallel with whatever the biology is doing, so we don't want it to be interfering with the biological reactions. We don't want the biological reactions to be interfering with the chemistry that we're trying to do. And so we're going to be adding things that aren't normally used as functional groups by the cells. So that's a basic overview of how we define bioorthogonal chemistry. And now let's get into some of the major advances. So in 1997, Carolyn Bertozzi published this paper in Science that a method they originally designed that was using ketones. This isn't like a truly bioorthogonal because our bodies have ketones, but they don't have this type of ketone that she was going to add in the position where they normally have it. And so we have some ketones like inside of our cells and stuff, but we typically don't find this group where you have a carbon double bonded to an oxygen, so a carbonyl with a carbon on either side. This is called a ketone. It can undergo various reactions, but typically those reactions are happening inside of cells, and you don't have this sort of ketone modifications on the cellular surface. So if you could introduce one of these ketones on the exterior of a cell, then you could specifically label those cells. Was able to introduce this into the sugar chains, the glycans that naturally coat the surface of cells, by feeding the cells a precursor sugar molecule that actually had a ketone added on. And so the cells would take up this sugar precursor, this individual subunit, incorporate it into those long chains of sugars, those glycans on the cellular surface. And now she could add a chemical, like their second reactant group, the matching reactant group. And this is actually going to react with that ketone to form this bond. And therefore, if you have that second thing you add to be labeled with such a dye, well, now you would be able to image things. If you have that second thing added to, say, biotin, then you could have this molecule called biotin, then you could add a molecule that can bind to biotin, so this avidin. In this case, they had this avidin conjugated, so this is attached to ricin A chains. This is like a toxin. They could then get the cells that were overexpressing or making too much of the sugar chains, such as cancer cells, to actually make this version incorporating the biotin, then add this avidin ricin side chain conjugate to actually get these cells to be targeted by a toxin. But that wasn't good enough. And so what was the problem? Well, the problem was that although we don't have those ketones on the outside of cells, we still do have the ketones inside of cells in various places. So it's not truly bioorthogonal, and there are various side reactions that could occur. And if you wanted to have super duper specificity, you would want something that wouldn't react with any biological molecules. And you'd want something too, ideally, that you can actually use inside of cells and other places other than just on the cell surface. And so they designed this second version. And this was going to use a modified Stoutinger reaction. And here they talk about this idea of chemical orthogonality. So the basic idea is that there's this Stoutinger reaction where you have this phosphine and this azide. So don't worry about these chemical terms. But you have these two chemical groups that can come together to form this bond. And this is a typical Stoutinger reaction. Now, this reaction, this product, the product that it forms, this bond, is not very stable in the presence of water. And as I mentioned, water is everywhere in cells and in bodies and in all of these things. And so if you have something that's not stable with water, that's a big problem. And so they developed a way in which you could do this reaction, but actually make a more stable bond. And this enabled them to use these azides instead of the ketones. They use these like 3 nitrogens in a row instead of having to use a ketone. And this allowed them to have more versatility in things like this. They can add various reacting groups to add different modifications. Similarly to how we saw before, they're adding this on to using these sugar precursors. Okay, but that wasn't good enough because, well, the phosphines weren't very stable. They had like oxidation problems, which I'm not going to get into. Various things with pH. Basically, it wasn't ideal. And additionally, you don't have as good of efficiency. So the Stoutinger reaction isn't perfect. And this is one of the things that like that Meldal and Sharpless were kind of like improving on. Was you could find a sort of similar reaction where you have these two things that can interact with one another in a specific way, but by making it so that these reactions are going to be more ideal. And it really, really crucial was this finding that you could add copper on this metal and make this reaction occur really fast and really specific. So this announcement, this was coming about in the early 2000s. Let me get my timeline. So this was happening in like the early 2000s. And Bertozzi had like, she was up on the literature and all this. She knew that this was existing. And she thought, okay, well, how can I incorporate that knowing that copper is going to kill these cells, kills these organisms? I need a version without copper. And by reading through the literature, she was able to discover some hints that you could actually use a strain-promoted click chemistry. So instead of relying on the copper to make the reactions happen fast, what you could do is you could actually use a molecule that is more reactive on its own without needing the copper. And so you can kind of see this alkyne, so this triple bond state carbon is in this weird ring where you have this ring strain. So it's kind of like uncomfy. And so this is going to make it more reactive in a reaction that is going to make it more comfy. And so this is going to allow this reaction to occur without the addition of copper. And so this was a really important insight and really important step forward for the field of bioorthogonal chemistry. And then this has enabled all sorts of further applications. So as I mentioned, there's this really great review article by Bird et al. Kind of detailing some of the various things that you could do with these reactions. So there's various other versions as well. You can see that this SPAAC, so this strained reaction that Bertozzi introduced, is really taken off and been really important, as well as other reactions that can occur. But you can see that these can be done to incorporate modifications to all sorts of different molecules, predominantly proteins in some cases because it's like easier to add specific modifications in specific places, including such as with the use of non-canonical amino acid incorporation, the split peptides where you can actually add part of it, like synthetically make this peptide to make this protein part and like stitch it together. Using enzymes to add the modification, like the precursors, various things like that. So the methods are better developed for proteins, but the modifications can also be introduced into other molecules. And it tells you more about how these various modifications can be added, various tags, types, and reactions. So this is a really just helpful review article. I'm not going to go through it all. I just wanted to show you some of the things in it, and so that you can see how it can be used in all sorts of different ways. And to just kind of like encourage you to check all this out, because I think it's really, really fascinating. You can use it for various things with drug development. There's also like click to release. So you actually have these click reactions be kind of releasing the active version of a drug. So very, very cool stuff. I'll also post links to some of Bertozzi's key papers. And so that's the one we're just on, as well as some more general audience papers. So this is a nice review from Jamie Durrani from Chemistry World, and then the Nobel Prize. You can actually watch the announcements. It's kind of fun to watch. They have a quick interview with Bertozzi. They have their popular science background on the awards, as well as the advanced topic. One of the cool things is it actually cites like the PhD thesis of one of her students. So very cool. As well as just some various articles from Bertozzi, some nice review articles she did. And yeah, so just basically there's a lot of resources out there if you want to learn more. And I highly encourage you to check them out if you want to learn more, because there's some really cool stuff. And basically the possibilities are just endless, because you can add so many different groups. And you can add these things that wouldn't naturally be possible, and that wouldn't be possible in the lab, like outside of the cell, and it wouldn't be possible without help inside of a cell. And really the two coming together is really the basis of this field of bioorthogonal chemistry, being able to introduce these precursors that then you can modify using all sorts of different modifications, taking advantage of just sticking like generic type of modifiers on one thing and another thing, generic in the sense that you can stick them anywhere, but super duper specific in that they're only going to react with one another. So you can stick them onto different things and get them to react together. So that kind of sticking the groups onto different molecules, allowing them to react together, is the basis of like click chemistry. The canonical click chemistry that is normally used, like talked about, is this like copper mediated cycloaddition. But click chemistry, that term can also be used for other things, other types of reactions like that. And then the idea with bioorthogonality is that you're actually getting these sort of reactions to occur on biological molecules. And really mixing and matching the best of biology and chemistry in order to add custom modifications to molecules, to track those molecules, to target those molecules, various really cool things that you can do. And so I hope this helps you understand bioorthogonality, the Nobel Prize and Carolyn Bertozzi's work. And so, yeah, so I was just really excited when I heard the news, and I wanted to kind of tell you all why it was so exciting. So I hope that helped.